Okay, good morning everyone, and welcome to the 2018 Royal Tyrrell Museum Speaker Series. To kick off the new year, the Royal Tyrrell Museum and its cooperating society are proud to present Mr. Jason Pardo. Jason is a Ph.D. candidate in the Faculty of Veterinary Medicine at the University of Calgary. Jason was born in California but grew up in Pittsburgh, Pennsylvania. He received his bachelor's degree in the Department of Environmental, Population, and Organismal Biology at the University of Colorado, Boulder. For his senior undergrad thesis, he studied the morphology of fossil lungfishes from the Permian and Jurassic of North America, as well as the genetics of tooth development in zebrafish. He subsequently moved to Alberta to pursue his master's degree in the Department of Biological Sciences at the University of Calgary. For his thesis, Jason used a micro-CT scanner to study the morphology and relationships of primitive amphibians called lepospondals from Kansas and Nebraska. After completing his master's thesis, Jason decided to stay at the U of C to pursue his Ph.D., for which he is studying the cellular biology basis for tooth regeneration in the axolotl salamander. Jason's research interests focus on the interplay between developmental biology and macroevolution in the origin and diversification of vertebrate body plans, particularly among early tetrapods. Over the years, he has published numerous articles on the early radiation of amphibians and tetrapods. Today, Jason will give us an overview of his research on early tetrapod evolution. So without further delay, I present you Mr. Jason Pardo.
Thank you, Francois, and thank you to the Cooperating Society for inviting me. So I'm going to be talking about tetrapods, and tetrapods are the group of modern vertebrates, modern bony animals with four legs, or that ancestrally had four legs. And if you look at this frog, this bright green frog on the lower right-hand side of the screen, you can see that it's got four legs. It's got two arms and two legs. And if you think about those frogs live right at that interplay, that place of interplay between land and water. So you're not likely to find a frog sitting in the middle of the desert. You could, but you're not likely to. But you typically are going to see them right on the shores of ponds and lakes and streams. But frogs are part of this group tetrapods that includes us, includes dogs, includes all the beautiful dinosaurs at this museum. And all of us together have one thing in common, which is that we share an ancestor that crawled out of water onto land about 370, 380 million years ago. So this is maybe not our direct ancestor, but this is a relative. This is an animal called Tiktaalik. This is the classic fishapod, your inner fish, from the Canadian Arctic. And you can see this is a very, very different animal than any of those creatures I just talked about, frogs or us or those dinosaurs. And we've now amassed this really, really good story of how we go from those fishapods through this diversity of forms in the late Paleozoic, so spanning that 380 million years ago until the end of the Permian at 260 million years ago. And then eventually resulting in modern diversity. So those amniotes, those lizards and mammals, as well as birds, as well as dinosaurs, on one hand, and then amphibians, so frogs, salamanders, and caecilians, which are these things that you probably have not seen, and I'll introduce you later. But going through this great diversity of forms in that late Paleozoic. The problem, and we've got this relatively good story here, especially at that fin to limb transition, of animals that are fishy, and then slightly less fishy, and then slightly less fishy, and more terrestrial over time. But these animals are relatively large. They look kind of like a cross between a salamander and an alligator, and mostly lived right on those shorelines. And they all look pretty similar, right? You probably could not tell the difference, except for the fact that some of them have fins and some of them do not. You would look at these and say, oh, they're all pretty much the same animal. And through this, we've kind of assembled a story of what's happening in early tetrapod evolution. So we have an initial period of time that's that fin to limb transition. So this is the period of time where we start getting limbs, we start getting fingers, we start being able to walk on land. that's going to be interrupted twice by mass extinctions. So there's 2 mass extinctions in the late Devonian, one in the middle of the late Devonian called the Kellwasser, and then one at the very, very end of the Devonian called the Hangenberg. And we know that these affect the vertebrate animals, the bone-bearing animals in the fossil record, because we end up, before this extinction, we have all of these shelled fish, like these placoderms here from Quebec, and then afterwards we don't. So after that extinction, then we start seeing diversifications. And this is the origin of those modern lineages. So the origin of the line that becomes amniotes, that's us plus dinosaurs plus all the descendants of our common ancestors, and then amphibians on the other hand, frogs, salamanders, and their common ancestors. And then we have a final period of terrestrialization and experimentation. So once we have those lineages, once we have amniotes and once we have amphibians, then they're going to diversify and they're going to start filling those terrestrial niches. The problem with that story is that if we go back to this, what we actually find is that that big diversity is a huge black box. We don't know how these animals are related to each other, and because we don't know how they're related to each other, we don't know what that means for the diversification of these groups, and we don't know what that means for the origin of amniotes and what it means for the origin of amphibians, particularly the origin of amphibians. And if we go to the amphibian fossil record, we find that it's really, really, really bad. It's okay from the mid-Mesozoic on, so when you get your classic dinosaurs, the amphibian fossil record's pretty good, but the amphibians all look pretty modern. When we go further back, it's really, really bad. So to give you an idea, there are 3 fossil frogs from the Triassic. So this is our frog transitional group. There's 3. We go to salamanders, the transitional lineage that leads to salamanders, maybe 3 or 4. We have 2 good ones. We go to caecilians. These are these long limbless guys that live in tropical rainforests today, look kind of like worms. We've got 2 fossils from the entire Mesozoic. Right? So this is, if you think about this, this is from the very beginning of dinosaurs to the very, very end of dinosaurs, we have 2 fossils. We know very little about these groups. So we've got ideas where these animals may have come from. And we have 2 major groups of animals in the Paleozoic that we can look at and say, maybe this is where modern amphibians came from. So on the left, you have lepispondals. This is a really, really diverse group of animals. They're all mostly very small animals. And then on the right, we have temnospondals. This is a more size-diverse group. Some of these are very, very large. Some of these are very, very small. Both of these are relatively diverse groups of animals, though. And they're differentiated mainly by their vertebrae. So temnospondals have these divided vertebrae. So you have a long strip of material that's basically the same thing as your intervertebral discs. And then you have bones tacked on for muscle attachment. In lepispondals, you have a single solid vertebra, much like our own. And we have hypotheses based on these of how modern amphibians evolved. And we have a temnospondal hypothesis where we place temnospondals as the lineage that leads to modern amphibians. We have a lepispondal hypothesis where those lepispondals, those small-bodied animals that are highly diverse, become modern amphibians. Then we have a polyphyly hypothesis where it's kind of a best of both worlds, or maybe worst of both worlds hypothesis, where caecilians evolved from lepispondals and frogs and salamanders evolved from temnospondals. And as we go into the fossil record, we can start to test this. So this, for example, is an animal called Gerra Botrytis that was described by my Ph.D. supervisor, Jason Anderson, back in 2008. And this animal looks very, very frog-like in some ways. It looks very salamander-like in some ways. And this animal got nicknamed the frogamander for exactly that reason. There's a reconstruction here on the right. And if we look at this and we compare it to modern frogs and modern salamanders, you can kind of make out how you could derive a frog skull or a salamander skull from the skull of this fossil animal. And if we look at animals related to the frogamander, so this is an animal called a brachiosaur. So it's called a brachiosaur because they have these external gills. And you find hundreds of thousands of these in fossil lakes in Germany and the Czech Republic. And you can look at this, you can find evidence of metamorphosis, of salamander-like development. So we're pretty confident, biologically that these animals are, in fact, the lineage that leads to modern amphibians, or at least modern frogs and salamanders. But the problem is that even though we're pretty sure that's right, by every measure that we use to actually test that, we can't differentiate between those two hypotheses, not to a meaningful level. And furthermore, these two hypotheses are still picking up on something that's biologically important. So what we wanted to do is we wanted to look at those lepus bundles in a little bit more close detail and maybe be able to say something about those deeper, more important biological features. So I'm going to, and again, these are all characterized by those single solid vertebrae. This is a tiny vertebrae. This vertebrae is only about 1 millimeter wide. And I'm going to focus on this group here first. These are a group called microsaurs, and then I'm going to talk a little bit about some of these other groups as well. So microsaurs are very small. The name is exactly as you'd expect. These are very small animals. They're thought to be kind of reptile-like in general body form. And what we've done is we've used micro-CT. So the micro-CT is a medical-based imaging tool. We take X-rays from 360 degrees around an organism, and then we use those X-rays to reconstruct cross-sectional data in 3 dimensions of where different densities of bone would be. And then we could take that, we could take a fossil, such as here, we can toss it in the CT scanner, and we can then go through there and digitally separate all the bones out, and then we can digitally separate specific bones that we're interested in, such as these delicate bones surrounding the brain. And what we found looking at microsaurs is that they're all kind of variations on a theme, but those variations themselves are pretty interesting. So you can see here a lot of these animals have sort of these pointy snouts, or they'll have sort of broad, sort of spade-like snouts, or maybe almost plow-like snouts. And but on the inside, they all look, again, pretty similar, but those skulls are specialized in different ways. So the structures here are all the same, but some of these bones are fused up. And some of these bones are modified into pillars, into structures to support the skull roof against the brain case and to protect that brain from pressures and compression pressures. And but in terms of the overall similarities, they're very similar to this animal up here, which is a reptile. And if we actually compare them side by side, they actually look very, very similar, even though the microsaurs themselves are very, very small. So we're pretty comfortable with that these animals are at least very reptile-like, but those weird morphologies also seem to align with modern reptile diversity. So I'm not saying these animals are part of that modern reptile diversity, but rather that they're doing the same things. So we start seeing these shovel-like snouts, much like some of these burrowing vertebrates. We see more sort of bullet-like snouts, like other burrowing reptiles, or maybe these plow-like snouts, like even more burrowing reptiles. So these animals all on this side are all amphibians. These are blind, limbless, burrowing lizards. On the right, on the left over here, these are all those microsaurs, and they all seem to align pretty similarly. And in fact, lizards are really good at doing this. Lizards are extremely good at adapting to dig through sediment and live underground. So we see over and over again, a few of these are snakes, but most of these are not. We see these plow-like skulls for digging through dirt. We see elongate bodies for, again, digging through dirt. Reduced limbs where present, or maybe tiny little remnants of limbs, but there's not much left there. So these are actually, in general, look a lot like those microsaurs. But when we compare to other lepus spondylus, we see a very, very, very different pattern. So there's that microsaur. This is an aistapod. This is an animal that's extremely elongate, almost snake-like or eel-like. These animals have between 120 and 180 vertebrae. These are extremely long, no legs at all, and very, very specialized open skulls. So this animal here specifically is an animal called Lithyscus. This animal is the earliest limbless tetrapod known. This animal is 330 million years old, from northern Scotland. And when we start peeling it apart, we find an animal that's very, very different from those microsaurs, but also very, very different from all known tetrapods. So we find, for example, here, this light blue structure here is actually part of the spine. It comes into the skull. So as I was saying, that squishy spine that a lot of these early tetrapods had, that's made up of the same material as your intervertebral discs, is actually extending way into the skull of this animal, which is something that you see in fish. You don't see it in modern tetrapods at all. This dark blue knob right here on this sort of descending gray thing, that descending gray thing is the space that would enclose the pituitary gland. And this little blue spot here is where the pituitary gland actually communicates with the mouth, which is, again, a very fish-like thing, a very primitive thing, and it's not something that we see in any modern tetrapod. We also see a division in the middle of the skull here that separates out the back of the skull from the front of the skull. Again, that's a very fish-like feature. When we put these animals into a phylogenetic analysis and take all these characteristics and let the computer work with them, what we find is that those aistopods are actually part of that initial diversification of tetrapods deep in the Devonian, deep within that fin to limb transition. We think that they were probably part of those animals which actually had digits and then must have immediately lost them, but they may even be further more basal than that. Those microsaurs, again, seem to be up as part of an amniote radiation and are not really closely related to aistopods at all. So we have to ask, how experimental was early tetrapod evolution? If we have at least two cases where these animals are doing very, very different and new things in very, very different parts of the early tetrapod tree, what about the rest of the lepospondylus? And when we start looking at, so potentially we could be wrong, maybe our tree is just wrong, but when we start looking at details of these animals, they actually do look relatively similar. We had sort of a narrower back of the skull than the front of the skull, some other features of the skull here that all seem to sort of align. And you can actually make out how you could take one of these very, very early fissiopods and turn it into an aistopod. You can't really do that. You can't really turn it into a microsaur, though. So we decided to look at those lepospondylus more broadly, and maybe does this actually work or does this not? So again, we can move microsaurs over to amniotes. We can move these animals here, Lysorophians. They end up being microsaurs themselves, and aistopods are part of that stem tetrapod radiation. But the rest of these we don't really know. But as we start looking at them, what we find is they also look very different. So we have these boomerang-headed guys, but on the inside, they don't look anything like any of these other animals. They all look very, very different. So what we think is actually happening is we have that initial fin to limb transition that we actually do know pretty well, but we were totally wrong. Experimentation happened really, really early, and it happened really, really often. Then that experimentation leads to the origin of those modern lineages, modern amphibians, modern amniotes. But that's actually part of that experimentation as opposed to the cause of that experimentation. And that lepospondylus are actually where all that experimentation is happening. They aren't a real group, but we've sort of clustered them together because we didn't know what else to do with them. So it kind of leaves us with a temnospondyl hypothesis, because lepospondylal hypothesis doesn't work. We don't have a coherent lepospondylal hypothesis of amphibian origins, and most of what we're seeing that are actually making lepospondylus is either convergence or just small body size. The problem is that temnospondyl hypothesis that we're stuck with doesn't really provide a good model for understanding caecilians. So caecilians, again, are those long, tropical, legless amphibians. They live in the dirt, under leaf litter. They eat worms and ant larvae and so on and so forth. And as my supervisor wrote in a 2008 review, the problem of list amphibian origins is really the problem of caecilian origins. And they're weird. These are really weird animals, and that's why it's such a difficult problem. And if you think this animal looks like a space alien, you're probably not alone, because you've probably seen one of these before when it tries to eat the Millennium Falcon in the beginning of The Empire Strikes Back. And so this is a very strange animal. It's got a tiny eye. It's got this big hole in the right anterior to the eye where there's actually a tentacle that they use for smelling. The rest of the skull is highly, highly ossified. You've got giant teeth, and this animal is very small, but big teeth. And again, no legs whatsoever, and the vertebrae all look the same. Problem is, when we go to the fossil record, they also look very, very weird, and we can't place them well within that Paleozoic radiation. So again, we have a small eye, maybe the beginnings of a hole for that tentacle. All the bones are beginning to fuse up. This just does not look like the things we see in the Paleozoic at all. So this is one of those few situations where I think we need to go out and look for new fossils. Maybe looking at the fossils we have just isn't solving that problem. So I and some collaborators have been working in Colorado for the last about 10 years now in an area in the western slope called the Eagle Basin. And these are equivalent to the red beds of New Mexico and Arizona, like Petrified Forest, Ghost Ranch, these very traditional, typical late Triassic fossils where you get animals like Coelophysis, some of those early dinosaurs. And we have some beautiful fossils from here. So this animal here is one of those armadillo crocodiles, an Aedosaur. But we also get these weird burrows that go all through these sediments. And this is my collaborator, Adam Huttenlocker, from the University of Southern California. He's measuring one of those, and the burrow's right here, and it kind of snakes down along that cliff there. And in those burrows, which have very, very specific morphology, we get these beautiful little skulls of these little amphibians. And these are tiny. These skulls are only about 2 to 3 centimeters long. We've got some that are even smaller. But when we toss them in a CT scanner, we get quite a lot of information, regardless of the fact that they're super tiny. We can start looking at the morphology of the eye. They have a nice ear. They've got attachments for the muscles of the neck. They've got fusion of different bones. And in a lot of ways, they look like early caecilians might look. We tossed them into a phylogenetic analysis with a lot of this information, and we found that our animal, which is reconstructed here by Jorge Gonzalez, we have our animal down here with caecilians, but it's quite far from salamanders and frogs. So salamanders and frogs are in there with those frogamanders that we always thought were the origins of amphibians. We're pretty confident with that. But caecilians are part of this group here that are a major Triassic radiation that we thought was a dead end. And these are a group of animals called stereospinals. And this is suggesting that maybe there's this deep divergence here of amphibians. When we compare our animal with stereospinals, it makes a lot of sense. So we have fusion of the base of the skull. We have very similar structure of the palate and of the skull roof. They look relatively similar. But it also looks a lot like the fossil caecilian. That's that Eos caecilia I showed you earlier. There are a number of bones that are fused. The structure of the palate, again, is very similar. And the structure of the base of the skull is very similar. Eos caecilia is more caecilian-like, but they all share some commonalities. So instead of the previous story where we had those frogamanders and then we had caecilians and we had nothing in between, now we have a really complex story here, but a very, very almost satisfying one, where we have a stepwise acquisition of more and more caecilian characteristics throughout the Triassic. We're still testing this. There's still a lot more to say. We proposed this in a paper last year, but we'll see how additional finds support or disagree with that. But as we go out and we look for more fossils, we find fossils that seem to be consistent with that. So this is a new animal from the Triassic of Wyoming. And these animals are found in these burrow horizons. So this is one burrow. This is from a CT scan. But you find horizons of dozens to hundreds of burrows. And in each one of them, you have a nice, beautiful little amphibian. And these animals are a little bit more primitive than our animal from the Chinle. These are a little older, too. But they seem to be telling the same story. So if we come back to this big black box here, we can maybe say some things about what's going on in there. So instead, we do have that story of that fin to limb transition, but it's more complex than that. There's a huge amount of innovation, and that innovation's happening at small body sizes, primarily within those lepospinals, but also in those temnospinals and in modern amphibian groups as well. That innovation is successful. We are seeing animals trying out new niches. Those new niches are things that we see later on tried out by new groups. Various lizards try out these same niches later. So they seem to be persistent. And this innovation seems to be associated with the origin of modern groups, modern amphibians, modern reptiles perhaps, maybe even the modern tetrapod group itself. And just to finish off, I'd like to bring us back to some stuff that Darwin said. Darwin, at the very end of Origin of Species, invites us to contemplate an entangled bank. This is the beginning of the endless form's most beautiful quote. But he asks us to imagine that all these organisms that are living on that tangled bank of a river or of a stream have all evolved through similar processes, but doing different things, trying new things out. And this idea of a tangled bank has become a metaphor in evolutionary biology for periods of experimentation and keeping diversity in order to experiment. And I'm obviously not the first one to apply this to the early tetrapods. It's an obvious analogy for early tetrapods. Matt Friedman talks about this in 2009. But what I want to finish is that these small-bodied animals, these lepospondylus and also modern amphibians, are probably part of this experimentation. This experimentation happens early, it happens often, and this experimentation is not some sort of side project in tetrapod evolution where what we really are interested in is that fin to limb, and once we have limbs, then that matters. But this experimentation is driving the whole process. It's driving tetrapods into new niches, into burrowing, into limblessness, and it's leading to those modern lineages as well. And that maybe we shouldn't be worried about losing lepospondylus or whether lepospondylus or temnospondylus are the right group, but rather that the entire story of tetrapod evolution is one of experimentation. With that, I have a number of collaborators that have contributed to this work. I obviously did not do all this by myself, and I have a number of people to thank that I don't have a slide for, unfortunately.